Hello everyone, my name is Alexander Spradin. I'm from the Federal Center for Animal Health of Vladimir, Russia, and today I'm going to talk about the epidemiology of lumpy skin disease virus, namely recombinant lumpy skin disease virus in the period from 2015-2020 with focus on the emergence and spread of virulent recombinant lineages. Lumpy skin disease virus belongs to the family Poxvirida, large DNA-containing viruses. This family includes viruses infecting vertebrates and insects. The family Poxvirida contains many genera, one of which is the genus Capripox virus, to which sheeppox virus, goatpox virus, and lumpy skin disease virus belong. Capripox viruses are brick-shaped, enveloped and double-stranded DNA viruses with genomes approximately 150 kilobase pair in size. The Capripox viruses are closely related antigenically and therefore cannot be distinguished serologically. They are also difficult to distinguish morphologically from other poxviruses, namely orthopox viruses. And the outbreaks caused by these viruses in sheep, goats, or in cattle are categorized as notifiable disease. Let's now look at the symptoms that Capripox viruses cause. In the past, the names to the viruses were given after the animals they infected. However, the symptoms that these viruses cause are shared and very characteristic. They are all characterized by the development of pox lesions on the skin. Sheeppox and goatpox infections may also result in the formation of lesions in internal organs such as the lungs. However, this can also take place in cattle. In sheep and goat, skin lesions begin as macules that harden to form papules, followed by eruption and necrosis. In the last decade, lumpy skin disease has consistently spread northward from its historical range of Africa into Eurasia. LSD cases were reported in Azerbaijan, Turkey, Europe, Balkans, China, India, Bangladesh, Vietnam, Sri Lanka, and so on recently. However, the first introductions into the Northern Hemisphere occurred in 2015-2016 when LSD was swept across the Caucasian region and the south of Russia, causing more than 300 outbreaks in 16 regions. To combat the disease, only sheeppox-based preparations were used. However, in 2017, the special pattern of genotypic nature of circulating isolates started to dramatically change. Unique vaccine-like recombinants emerged that were composed of Neethling virus strain and vaccine-kin and KCGB virus backbones. No genetic input from the field isolates were identified, which clearly demonstrates that these were incursions from outside, which I will talk on later. You can now look at the distribution of outbreaks in the period 2014-2016. The virus spread into Europe, Turkey, and the south of Russia. And the most important thing here is that all the outbreaks were caused by a single lineage, a field strain belonging to cluster 1.2. As I already said, in 2017, we started detecting strains that are not related to the previous outbreaks or to the vaccine used. These were recombinant, naturally occurring recombinant vaccine-like strains belonging to the Kenyan vaccine that was used in neighboring regions of Kazakhstan. Owing to the use of the shippox-based vaccine, we were lucky to detect the strains, the recombinant strains, because shippox-based vaccine preparations do not cause any clinical signs, or no DIVA strategy is required to detect LSDV. So at the interface of the different vaccines, we detected Kenyan lumpy vaccine strain genetic signatures in outbreaks caused in the Russian regions. The presence of lumpy skin disease virus was confirmed following an initial PCR-RC analysis targeting the conserved P32 gene fragment of capripox viruses. Subsequently, the sample was subjected to different PCR assays, differentiating between vaccine-like viruses and wild-type field isolates based on open reading frame 8 of the vaccine strains and open reading frame 1 to 6 of field isolates. So the results were as follows. The strains were positive for the capripox virus level, negative for vaccine strain DNA, and negative for field strain DNA. As a result, we were able to identify a novel, naturally occurring recombinant lineage formed by two vaccine strains, a Nithling vaccine strain and Kenyan vaccine strain. This tree demonstrates the phylogenetic affiliation of the novel strain. Although it forms a sister lineage to the vaccine group, it's not identical to the vaccine strains, to the commercial vaccine strains, nor it belongs to the field strain. In 2018, our outbreak spread along the border towards the Far East, very close to the border with Kazakhstan. All the outbreaks in 2018 were caused by recombinant strains. However, it's important to note here that no live vaccines were ever used in Russia. Let's now look at the phylogenetic tree representing the current genetic variation of available sequences. The tree shows that the isolates detected in 2015-2016 belong to the field cluster. However, all the isolates detected in 2017 and onwards all belong to recombinant lineages. Let's now turn our attention to the OIE manual on lumpy skin disease virus. What is the definition of disease? Antigen or nucleic acid specific to LSDV excluding vaccine strains. This explains why there are no detection of vaccine-like outbreaks in other countries where homologous vaccines are used. Following this logic, all outbreaks in 2018, 19, 20, 21, 22 in Russia and other countries were exempted from being reported to OIE because the current PCR assay detect vaccine strain lineages. I often hear claims that we, from our European colleagues, that we didn't detect any outbreak caused by vaccine strains. Of course, there won't be any outbreak caused by vaccine strains because this is not a disease, and any detection is exempted being reported. This is the reason why the situation on the epidemiology of LSDV worldwide is underrepresented due to the this definition of the disease. However, vaccine strains do cause needling disease in the form of adverse side effects. But again, these symptoms are not reported or are not welcome to be reported. This is how the situation looks nowadays. The lumpy skin disease, when it occurred first in the Caucasian region of Russia, spanned about 9,000 kilometers across the border towards the Far East region. If we add Southeast Asia and South Asia to the map, we will see the distribution range of LSDV. However, this situation is tricky because if we look into the genetic backgrounds of outbreaks, the situation will look very different. It's important to know that the ongoing pandemic is unfolding in Southeast Asia due to recombinant strains. Special temporal clusters have been identified that are represented by individual recombinant lineages, showing that the clusters are not clonal. Statistical analysis shows the direction of spread. It comes from outside Russia into Russia. All outbreaks congregate along the border, and if again we include the genetic analysis, we will see that even outbreaks are not even related, they are not clonal. Outbreaks each year are caused by unique recombinant strains, which clearly shows that the spread of LSDV in Russia is not related to the previous outbreaks that were first reported in the Caucasian regions, but represented by incursions from outside. This is a map showing the epidemiological situation on lumpy skin disease in Southeast Asia. It's important to know that the first outbreak in China occurred very close to the border with Kazakhstan, about 25 kilometers from the border with Kazakhstan and 700 kilometers from the border with Russia. However, the first representatives of this particular lineage were detected in China, and then we detected this lineage in other regions of the Far East of Russia and Mongolia. These findings demonstrate that outbreaks in the Far Eastern regions of Russia cannot be considered as a source of this virus. Full genome sequences have been analyzed, and the tree was constructed. We can now see that the lineage circulating in the regions of the Southeast Asia is currently dominant, and all outbreaks that are caused by this lineage are related. This map demonstrates the distribution of different recombinant lineages across Russia and Eurasia. Each color designates a different lineage based on the available full genomes. While in Russia every outbreak was caused by a different strain, the situation in Asia is caused by two lineages clustered, 2.5 and KCGP. Here it's evident that there was no clonal radiation of the wild-type counterpart from Caucasus, Turkey, and Balkan. The epidemic was successfully eradicated in Russia with chickenpox vaccine. And from 2017 onwards, recombinant strains spilled over into the Russian territory from outside. So I draw your attention here that the epidemic with recombinant LSDV strains played out in Russia, where only chickenpox-based vaccine is allowed for use. In this scenario, Russia had nothing to do with the ongoing pandemic of LSDV in Southeast Asia. Thank you for your attention. If you have any questions, I will be happy to answer.